This module will be on DNA markers, and we'll be looking at types of DNA variation and the molecular and genetic techniques used to study them. So DNA markers are a relatively recent area of study in genetics, and as such, information is rapidly being overturned, nulled, or adjusted. And this is the result of growing sophistication of new technology used to investigate genetic markers. And as such, much of what's discussed herein may be dated quite quickly. However, a note, due to this field being a young one, there has not been much internal critical evaluation of published material. As such, it is always wise to look at studies with DNA markers with a critical eye instead of taking it for strict fact. Alright, so some general notes on DNA variation. So the variation being discussed in this module is variation in DNA nucleotide structure, not variation in alleles or genes. So variation is predominantly from so-called non-coding regions. These regions are sections of DNA between genes that do not encode for proteins. Likely that non-coding concept will be rejected in the future, as there is research suggesting that these regions do play a significant role, albeit an indirect role, in coding. Alright, so we're going to spend a lot of time talking about restriction fragment length polymorphisms. So these are variations in patterns of fragments produced when DNA is cut with the same restriction enzyme. So variation is typically the result of a single nucleotide base change. So restriction enzymes recognize specific sequences of target DNA. They cleave the DNA molecule across the sugar phosphate backbone at that sequence, creating two DNA fragments. Enzymes are named for the bacteria from which they originate, and if the enzyme cannot restrict or cut the DNA, there is a mutation at that restriction site. So when some individuals have a particular restriction, while others, due to mutation, lack that restriction, this is a restriction fragment length polymorphism. Right, another type of DNA variation are insertions and deletions, collectively referred to as indels. And this is essentially the insertion or deletion of nucleotide bases. Now, they are referred to collectively as indels because it is impossible without comparative studies to determine whether the deletion or insertion status is the original configuration of the DNA. Now, an example of this is a 9 base pair deletion. So this is located in the mitochondrial DNA, and the polymorphism is classified as a deletion as most individuals have the deleted sequence. So the sequence is as follows, as you can see on the screen. And if you look at the deletion sequence, it is missing one of the larger segments of nucleotides. All right, another DNA variant is what we call interspersed nuclear elements, signs and lines. Signs are short interspersed nuclear elements, typically 150 to 300 base pairs long. And the best studied sign is what we refer to as the ALU sequence. Now, longer interspersed nuclear elements are typically much larger, around 6,000 base pairs long. And polymorphisms are those in which different individuals have a different number and or location of these interspersed nuclear elements. Now, these elements are considered to be transposable. And what this means is that they are mobile DNA sequences that insert into many locations within a genome and often cause mutations and DNA rearrangements in the coding regions. All right, the final type of DNA variant are what we call tandem repeats. So these are repetitive sequences that occur at specific locations throughout the genome. Variable number tandem repeats, or VNTRs, are longer in length and as such, they have a lot of internal variation within the VNTR. And this variation is often unique to individuals, and as such is used as a DNA fingerprint. Now, short tandem repeats are shorter in length, and they can help distinguish between homozygotes and heterozygotes on autosomal loci. Both of these are commonly used in forensics investigations. So how do we actually study these DNA variants? So we have a tool called polymerase chain reaction, or PCR. This is a process of amplifying or making copies of DNA by a billionfold within only a few hours. Now there are 3 steps: denaturing, annealing, and extension. And the end result produces up to 1 million copies of DNA. So the denaturing step is when DNA is heated to an extremely high temperature, causing the 2 strands to separate. The second step is annealing, and this is when DNA is quickly cooled, allowing short single-stranded primers to anneal to their complementary strands. And then finally, we have extension. This is when it's reheated to a moderate temperature, which allows DNA polymerase to synthesize new DNA strands. All right, so after we have amplified our DNA fragments using PCR, we can use a tool called gel electrophoresis. This is a method of viewing or assaying DNA fragments. So it relies on the DNA fragment size and the inherent negative electrical charge to separate fragments. So essentially, it sends an electrical current through the gel. DNA fragments will run from the negative pole. Smaller fragments will travel farther and faster than larger fragments. So this is used to identify indels. So DNA fragments with insertions will be larger, while DNA fragments with deletions will be shorter. It can also be used to look at heterozygosity. Individuals who are heterozygous for an insertion will have 2 bands, while individuals who are homozygous for an insertion will have only a single band. Now, a final, more new method is what we call pyrosequencing. And so this is a modified DNA sequencing method, and it allows for rapid sequencing of large segments of DNA. So whole genomes can be mapped in months instead of years. And so in this instance, nucleotides have been chemically modified to emit a photon of light when bound to their complement on the strand. Measures of the light bands released for each of the 4 nucleotides can then create a nucleotide sequence map. So pretty cool new stuff.